Forensic entomology.
The study of the insects associated with a human corpse.
This type of analysis is primarily used to determine elapsed time since death.
However, insect evidence may also show that a body has been moved after death,
Or that the body has been disturbed, either by animals or by the killer returning to the scene of the crime.
The location of insect activity may provide clues as to the presence and position of wounds.
In court, entomology may corroborate other physical or testimonial evidence.
The science of forensic entomology can even be of value in demonstrating abuse or neglect of living children or senior citizens.
Most cases that involve forensic entomology are at least 72 hours old. Within 72 hours, other forensic methods may be more precise than insect evidence.
However, after 3 days, or in cases involving dismemberment, insect evidence is often the most accurate and sometimes the only method of determining elapsed time since death.
There are two methods used by an entomologist to determine elapsed time since death.
The first method looks at maggot age and development. This method is more likely to be used in cases when a body is discovered within a month of death.
The analysis of maggot age and development can determine a date of death which may be accurate to a day or less. Maggots are larvae, or immature stages of two-winged flies, often the first witnesses to a crime.
The first to arrive are blowflies. They lay their eggs in batches on the corpse, usually in a wound or in any of the natural orifices. Their development follows a set, predictable cycle.
An egg will hatch after a predictable period of time into a first-stage larva. The larva feeds on the corpse and molts into a second-stage larva. The larva continues to feed and develop into a third-stage larva.
The stage can be determined by size and the number of breathing holes. Second and third stage larvae are also easily identified by the red gut they develop.
When in the third instar, the larva continues to feed for a while. Eventually it stops feeding and wanders away from the corpse, either into the clothes or into the soil to find a safe place to pupate. This non-feeding wandering stage is called the prepupal stage.
The larva then loosens itself from its outer skin, but remains inside. The outer skin hardens into a protective shell or pupal case, which shields the insect as it develops into an adult. Freshly formed pupae are pale in color, but darken to a deep brown in a few hours. After a number of days, an adult fly will emerge from the pupa, and the cycle will begin again. The empty pupal case is left behind as evidence that a fly developed and emerged.
The analysis of maggot age and development can be used effectively only until the first adults begin to emerge.
After a single blowfly generation has been completed, the entomologist will then begin to look at the successional waves of insects. This method is generally used when the corpse has been dead for a month or more.
The human body supports a very rapidly changing ecosystem, which will progress from a fresh state to dry bones in a matter of weeks or months. Different stages of this decomposition are attractive to different species of insects.
Some species are not interested in the corpse when the body is fresh, but are attracted to the corpse at later stages in the decomposition process. Some insects are not attracted by the body directly, but arrive to feed on the other insects at the scene. Many species are involved at each stage of decomposition, and each wave of insects overlaps the next.
Therefore, with a knowledge of the insects common to the region and an understanding of when these different species will arrive on scene, the insects associated with the remains can be analyzed to determine a window of time in which death took place.
In order for any of this analysis to take place, there must be careful and accurate collection of insect evidence at the scene. This is best performed by someone with knowledge of insect behavior. I am always willing to attend the scene if possible. Unfortunately, I'm often not called until after the body has been removed from the scene. I may see the remains at the morgue, but in many cases, I may not actually see the remains at all. Under those circumstances, the quality of analysis will really depend on careful collection of insect evidence by the investigating officers.
Samples of insect